It gives me great pleasure to introduce a good friend of mine, John Rogers. John's going to talk about soft electronics for material, fetal, and neonatal health. John is an amazing researcher, and his background is quite interesting. Currently, he's director of the recently endowed Corey Simpson Institute for Bioelectronics at Northwestern University. He's co-authored nearly 900 papers and is co-inventor on more than 100 patents. John's background is BA and BS in physics and chemistry from UT Austin, and then advanced degrees at MIT, including a PhD in physical chemistry. And then he transitioned into industry. He was a member of technical staff at Bell Labs, which was an amazing period of time at Bell Labs, the last horizon at Bell Labs, but also was there with Zhen and Bao, our esteemed colleague here at Stanford. John then took up his first academic post at the University of Illinois Urbana-Champaign in 2003, launched an amazing career that complemented what Zhen's doing, but in a very different way. And then he moved to Northwestern University. And he's had many notable recognitions over the years, including the Lemelson MIT Prize and many other things. And he's an elected member of all three national academies: National Academy of Medicine, National Academy of Engineering, National Academy of Sciences, and he's also an elected member of the American Academy of Arts and Sciences. So with that, please join me in welcoming John Rogers.
All right. Okay, great. Thanks so much for that introduction, Joe. It's great to see you and to be part of this incredible symposium. I really enjoyed the talks this morning. And I'll try to share with you some work that we've been doing in this broader area of kind of research at the boundaries between engineering science and medical science, with a very strong kind of translational emphasis, mostly around diagnostic devices and sort of hardware development. My background is in material science and engineering, but I run a very interdisciplinary research group, including folks with expertise in electrical and computer engineering, chemistry, biomedical engineering, mechanical engineering, deeply coupled into our medical complex in Chicago, with joint appointments for myself in neurological surgery and dermatology. So I'll try to share with you today some technologies that we've been working on to address kind of needs associated with vulnerable populations. So real focus on beginning of life health challenges, specifically maternal, fetal, and neonatal health, because I think there's a sort of a gap there, and there's some opportunities for engineering advancements to really improve patient outcomes in those spaces. And so I'll give you a really quick background around the nature of the hardware and soft electronic systems that we're developing in this context, but not too much engineering. Really try to focus on the medical applications, hopefully a little bit more resonant with this particular audience. In biophysical monitoring, I'll tell you a little bit about that, but as Joe mentioned at the outset, I think there's a lot of value in liquid biopsies and trying to think about biochemical assessments of health status, and try to give you a sense of what directions we're trying to pursue in that space. So before I get started, I just want to sort of acknowledge, Sam has been an inspiration to the broader community here, and really, has had a tremendous impact not only here at Stanford, but on others, including myself. I didn't know Sam all that well, and I haven't co-authored papers with him, but I met him several times during visits to Stanford over the years, giving department seminars and so on. I was really struck by his energy, and maybe more importantly, by his kind of worldview and perspective around how we as academic scientists can really make a contribution to societal challenges in healthcare. And I pulled this particular quote as a former student of his that I felt it was really kind of touching and sort of resonant with my own views and perspectives around what we can do in engineering science to try to improve the world and improve the health of patients, and really thinking about scientific research in an academic setting, not where the end goal is exclusively publication in Science or Nature or Cell. We do plenty of that ourselves, but thinking about those publications as starting points for translational efforts to try to take those new ideas and really bring them and deploy them at scale, to improve the way that we do health care delivery, but not just here in the U.S., other developed parts of the globe, as I'll show you through the course of this talk, really thinking about cost-effective techniques that can impact everyone, including those in very resource-constrained areas, lower and middle-income countries where we have quite a number of activities. So I just want to acknowledge Sam and his vision, and just recognize its sort of resonance with my own aspirations for how I might be able to make a small impact in this broader space. And so we do that through an institute that we run at Northwestern. I think a lot of the same goals as those of the FIND center, as Joe outlined earlier this morning, but again with more of an emphasis on hardware technologies and things we can do in advances in electronic, photonic, microfluidic systems and devices that can kind of integrate with the body and provide capabilities that are currently available only in hospital setting at one level, but also thinking about going beyond and thinking about de novo measurement possibilities also, and again in cost structures that will allow access to anyone in any place around the globe. And so that's kind of what we do. Number of different activities within this institute. It's an endowed institute, and so it provides discretionary funding on an annual basis. It allows us to do sort of translational level engineering that goes beyond the kinds of things you can support with an NIH or an NSF grant. I won't go through all the various areas here, but just give you a sense. There's a broader scope than what I'll share with you today, but the two platforms that I will describe represent core activities within the institute. And one of them is sort of a soft or skin-like form of electronics, and many of you may be familiar, as Joe referenced, the programs in Janan Bao's group, colleague of mine from Bell Labs days, and we're interested in that kind of technology as well. Got started in it quite early on, but a little bit more from an engineering rather than a chemistry standpoint. And I won't go through the details, but we now know how to build sophisticated forms of electronics, physiological sensors, wireless communication platforms, power supplies that all can adopt skin-like properties, physical properties. So thickness is comparable to the thickness of the epidermis itself. Areal mass density is about the same. Thermal mass, mechanical properties, but all with the ability to sort of blend in high-performance inorganic electronic materials to build systems that adopt the form factor of a temporary tattoo, but with all the sophistication of electronics technologies that you would see in a more conventional semiconductor device, a microprocessor in your cell phone, for example. Now, the skin interface in this case adds a tremendous value because you can use that skin contact as the basis for doing measurements of underlying physiological processes to get a picture of an individual's health at ICU-grade level of quality, but without wires and with the ability to do that continuously in remote or home settings. So we kind of figured out how to do this, and again, I won't go through the details. A lot of engineering that's needed to make this happen, but we felt we had a pretty good handle on it in 2011, got a little bit better in 2014, published a few more papers, broader community got involved, and at that point we decided we had enough sophistication that we could actually begin to engineer these systems to match and meet unmet clinical needs. And the basic science here really is in hybrid materials integration and innovation, and thinking about materials and mechanics together with circuit design that allows you to put together full functional systems that have mechanical properties that enable this persistent, non-irritating, imperceptible interface with the human body, not just at the wrist and the finger like an Apple Watch and an Oura Ring, but at a relevant location across the body and anatomically matched to a particular measurement need, all with wireless capabilities with time-synchronized operation for multi-nodal deployment on patients where that's required. And so at this point, there's a broad portfolio of technologies that have that kind of characteristic, either ultra-miniaturized in form or skin-like in their mechanics, and engineered to really reproduce, as I mentioned before, sort of ICU-grade measurements of physiological health status, but without the wires and without the need for insertion into a controlled environment like an ICU or otherwise a hospital setting. And so that's kind of the status. If you think about it from an engineering standpoint, these platforms and these concepts are quite versatile. So a single device can include multiple types of sensors operating based on thermal or electrical, fluidic, as I'll describe in the second half of this talk, mechanical, optical, sounds, body sounds, mechanoacoustic measurement modalities. I won't go through all the details, but each device can incorporate multiple sensors of these various types. And as I mentioned before, multiple devices can be mounted on the body simultaneously for wireless time-synchronized operation of sort of full body characteristics of health status, where the goal in all cases is clinical quality where there is a clinical predicate, but also thinking about going beyond standard of care to novel measurement modalities as well, where continuous monitoring is a key feature of these technologies. Big data, we heard about machine learning, being able to feed those machine learning algorithms with high quality, multimodal, continuous data, but all engineered with a consideration around cost, deployability, so that they can be made available to everyone, not just people who are in privileged situations like we are in terms of access to expensive healthcare apparatus and hospital infrastructure. So that's the idea. If you have that technology, you think it's working, what sort of patients would you want to start with in terms of engineering for specific measurement requirements? And we decided that premature babies might be the best example, most compelling example of how these kinds of technologies could benefit patient care, eliminating the wires, eliminating the high-cost data acquisition systems that are needed to pair with those sensors connected by hardwired interfaces, eliminating the adhesive tapes that are needed to keep those wired sensors against the fragile surface of the underdeveloped skin on these babies, and thinking about getting rid of all of that old technology, replacing it with these wireless skin-like devices to really revolutionize the way that we deliver care in a NICU type setting. So this was a slide that we put together maybe 2016, 2015 or so, just a vision of what we had hoped to do at that time. This is just Photoshop, but it turns out that you can do all of that, and you can render all of those measurement capabilities in these full epidermal or skin-like forms that I referenced before. It's 2 patches, reproduces everything that's done as a standard of care, even a level 4 NICU. One device shown resting on the fingertips here goes on the chest for continuous single lead ECG measurements. There's also a very high precision digital temperature sensor, gives a pretty good estimate of core body temperature from measurement on the chest. That device pairs with a second device shown on the palm of the hand that interfaces with the limb, either a hand or a foot, to do photoplasmography for SpO2 measurements. And those 2 devices are wirelessly time synchronized, so you get some hemodynamic information in terms of pulse wave velocity that in some cases with a calibration can serve as a surrogate for systolic blood pressure. So we were able to get IRB approved use of those devices in an active operating NICU in Chicago, Lurie Children's Hospital. And so it did tests on about 200 premature babies coming through the hospital, parental consent. This is one of the devices on the chest, you can see it. But because at that time it wasn't FDA approved, you had to maintain sort of the FDA approved wired-based sensors in place as well. And so that turned out to be very advantageous because it allowed us to do one-to-one quantitative comparisons of the wirelessly collected data versus the wired-based measurements. Turns out the precision and the accuracy actually higher because you eliminate a lot of the motion artifacts associated with measurements that require those hardwired interfaces. So we're able to do that, like I said, about 200 babies, still ongoing. Lurie Children's Hospital, Prentice Women's Hospital, both in downtown Chicago. This is the youngest baby that we've examined, is 26-week delivery. 24 weeks is kind of the edge of viability, so this is getting pretty close to that time point. We're not software engineers, but we had to put together a sort of graphical user interface that replicates the kind of GE dash type interface that the nurses are accustomed to. So real-time ECG, SCG as well. We're doing cardiac sounds, and then the red and the IR channels of the photoplasmography that we're doing simultaneously, and then readout of heart rate, SpO2, pulse transit time, and temperature in that case. So that worked pretty well. The nurses really liked the devices. The parents also, the idea of getting rid of the wires and the invasive tapes, very, very attractive for parents who have premature babies in that type of setting. So we actually expanded the deployments out to include the pediatric intensive care unit, a little bit bigger babies in those cases, but still benefiting from wireless mode of operation. You see the chest unit there, so you're getting heart rate variability, respiration rate, core body temperature from that sensor, and you get SpO2 from this one. And because they're time synchronized, again, I just mentioned that you get the pulse wave velocity, hemodynamic measurements, and that's going beyond standard of care, but it's a very natural option with this particular setup. So this is kind of the raw data that comes out of those devices. There's the ECG, there's the 2 channels from the photoplasmography, there's cardiac sound, so you measure the opening and closing of valves, complementary data to that which you obtain from ECG. There's a high bandwidth accelerometer in the chest units. You actually get chest wall movements, and so another redundant measurement of respiratory rate. Other things as well, I'll mention that in a second. Now currently you take all that data and you just distill it down to very simple vital signs, heart rate. You get heart rate variability there as well, SpO2, respiration rate, temperature. But there's a tremendous additional amount of insight and information that can be extracted from these kinds of data. Mayo Clinic has demonstrated that you can determine ejection fraction from ECG, for example. So this is what the devices are doing. This is what clinicians are looking at, but there's a lot of additional information there that opens up opportunities for advanced machine learning techniques to be applied to that kind of data. I'll get back to that concept in a little bit. So we published these results, and then got quite a bit of attention in the popular press. That was spring 2019. Shortly thereafter, I got a call from program managers at the Bill and Melinda Gates Foundation who were paired and partnered with the Save the Children Foundation, asking whether we could adapt those technologies to allow for deployments into lower and middle income countries. And that occurred to us as a really exciting engineering challenge to think about how to do that and how to change the engineering designs to align with manufacturing infrastructure and to align with the kind of capabilities that are available in those remote resource-constrained clinics. But we were actually able to do that. So following that 2019 paper, we were able to put together an equivalent pair of devices that satisfy the cost requirements needed for deployment into places like India, Pakistan, Zambia, Kenya, Ghana. We're also in Mexico. This is through to 2022, but we continue to support those deployments currently. So that's kind of what it looks like and what we were able to do. This is an example of first deployment into Zambia. So there's a couple of my students here. These are healthcare workers in Zambia. I think I'm taking a picture here. I spent a couple of weeks in Zambia myself, but very, very powerful mode of utility in these contexts. He has a limited number of healthcare workers, and so being able to use real-time diagnostics and measurements of health status allow for a triage deployment of those limited healthcare resources. That's the real value proposition in places like Zambia and the other places where we're currently deployed. So that's about as far as we can push it as an academic group, but as I mentioned, we and this group is also very interested in translation, getting these things out at scale. So it was really kind of a forcing function for us to put together a very small, tight, lean engineering team to take the devices to the next level. So we have a number of FDA clearances, 510K clearances. They're just showing equivalency to predicate devices. Several thousands of sensors have been deployed, 20 active countries, still supported by the Gates and the Save the Children Foundation, but now also by Merck for Mothers and the Steele Foundation as well. So these are the kind of numbers, but it's all open source, so we publish everything about these technologies as well, and so that continues to be an important part of our approach to technology development. So a couple of milestones recently. So we demonstrated the first wireless NICU in a collaboration with Montreal's Children's Hospital. That was in October. We have financing in place, so that's no longer a constraint. And probably most importantly, we have partnerships with large companies that currently supply hardware into NICU and PICU facilities. This is representing about 75% of the global market in that space, Draeger, GE, and Space Labs, and those are all non-dilutive funded joint development efforts with those companies. So I think everything is on track. We'll see how it goes, but I think everything is lined up for successful deployment and translation. But then you can think about, like, what does the NICU of the future look like in terms of monitoring? So you have these devices, they're proven, they reproduce what's done today, but they have the capacity to insert additional modalities of operation. So we have that high bandwidth accelerometer in the chest unit, so you can measure activity, you measure body orientation continuously. And so one of the things that they push in LMICs is this idea of kangaroo mother care. They don't have incubators, so mothers holding their babies close to the chest turns out to be an important part of healthy care of babies at that stage of their lives. And we actually monitor the orientations. We know when the babies are in this kangaroo mother care, and we can sort of quantitate the beneficial effects on heart rate and SpO2 associated with that kind of care. Other things you can do is you measure vocal biomarkers. So you have this high bandwidth accelerometer sitting on the chest. You can actually measure vibratory signatures associated with crying. But it's not a microphone, so you're not confounded by ambient noises. So you're really just measuring crying of the individual baby that is supporting the device. So maybe crying time, tonality, a lot of data there. Maybe that could be added to the mix. Machine learning, maybe you could develop additional insights by looking at that kind of data. So that's just one example of technologies that are moving out of our institute into what I would say is kind of a translational pipeline, moving through FDA regulatory processes, moving through manufacturing and scale deployment. So let me just give you some other examples, not all of these, I'll just give you a couple of examples of things that we're working on. So I mentioned there's sort of the title is maternal, fetal, neonatal. Talked about neonatal, pediatric. What about maternal, fetal? Well, same kind of technologies can be used. Actually, pretty high mortality rate associated with childbirth in some of these LMICs. So Gates is funding this as well. And so can you monitor the health of the mother, the health of the fetus entirely through that full intrapartum period? And it turns out you can do that. You can just modify the devices I just described to you for measuring pediatric neonatal health for the mother. And so a unit on the chest, there's one on the fingers doing SpO2, photoplasmography. But now we have a third wirelessly synchronized device, so multinodal. Again, that kind of concept is important here. Mounts on the belly. It does Doppler and it measures uterine contractions, EMG, and body orientation as well. And all that's time synchronized. So we've deployed these into Prentice Women's Hospital, mothers giving birth. And there you can also do this kind of benchmarking against this traditional wired-based systems for monitoring health of the mother, health of the fetus. I won't go through the details, all published in PNAS in summer of 2021. But maybe a more exciting thing is that Gates funded us to deploy these again into LMICs, and this paper actually includes data from 487 pregnant women during delivery in Zambia. So you have devices set up, expecting mothers present to the clinic. They're mounted with these devices, and throughout the intrapartum period, you measure the vital signs associated with the mother and the fetus. But again, you get additional information, so you get positionality because you have the chest unit has that accelerometer, so we can sort of correlate body position with health status of the mother and the fetus. So you're beginning to sort of think about digital biomarkers associated with health during childbirth. And then you can think about like measuring an individual patient, but you have 487. Actually, 3 of these women passed away during childbirth. So don't still have enough statistics around how to identify signs that could lead to mortality, but you can see how this would go in the future. It's all common format in terms of the data. And maybe 10 or 100 times more women, moving through studies of this type, you might be able to determine not only the individual's health status at a particular time, but like make predictive assessments of health trajectories, where things are likely to go, when birth is likely to happen, whether complications are likely as well, by really using this population scale data and machine learning. And so I think this is a very, very important future, and one that Gates is interested in as well, is using this kind of digital data to improve healthcare. So anyway, another example. Let me give you one more. And in this case, we're not just reproducing measurements that are done in the hospital today. This is de novo measurement capabilities that flow as a result of the unique engineering features of these soft skin interfaced electronics. And that is in measuring flow of cerebrospinal fluid in pediatric patients who suffer from hydrocephalus and have a shunt inserted to treat that condition. So probably many people in this audience are very familiar with the condition. Hydrocephalus is associated with the overproduction of cerebrospinal fluid that can create very high, abnormally high intracranial pressures that can be life-threatening. It could be congenital, can be also brought on by traumatic brain injury. Typically the way that condition is treated is you put a tube in the ventricles of the brain, there's a check valve, and then the tube moves into a distal location of the body to relieve the excess pressure as necessary. Works really well, but you have almost a 99% failure rate within 10 years after the shunt is installed, because the shunt can become clogged either by kinking or obstruction. It could rupture. And the problem with this is that it leads to nonspecific symptoms. So if you have hydrocephalus, you have a shunt inserted, you develop a headache or you begin to feel nauseous, you don't know if you just have a headache or you ate something that disagreed with you, or your shunt is clogged. If your shunt is clogged, it could be life-threatening. If you just have a headache, it's not a problem. So if you have this condition, it has a huge detrimental impact on your lifestyle, because every time you have a headache, you have to go into the hospital to have your shunt checked. And that involves CT scans, X-rays, all kinds of very invasive, sometimes surgeries, and so on. This is one patient that we worked with early on. And so how could this kind of skin-like electronics address this patient need? Would there be a way to develop a device, you just put it on the skin and tell you if your shunt is clogged or not, would tell you, the flow rate of cerebrospinal fluid through the shunt as an additional sort of capability. It turns out that it's easy to do that because you have an intimate, low thermal impedance contact with the surface of the skin. Your device itself has very low thermal mass, and so what that means is you can track very subtle changes in temperature at the surface of the skin to about 10 millikelvin precision. So if you couple that very sensitive temperature measurement capability with a thermal actuator, so an ability to launch heat, thermal power into the surface of the skin in a very controlled amount, you can surround that thermal actuator with these very high precision temperature sensors to measure the distribution of temperature that results from that local heating. And if you position that device over a blood vessel, or as I'll show you in a second, a shunt, what will happen is the resulting temperature distribution gets skewed in the downstream direction, as you might expect, just due to the thermal effects of that directional flow. So you actually measure that degree of anisotropy and use computational modeling techniques to extract out or predict what the flow rate must have been in order to reproduce that kind of thermal anisotropy. So it turns out that that works really well. You can make the measurement in about 2 minutes, and you can get a very accurate measurement of flow of any type of vessel or shunt or tube that is sufficiently near the surface of the skin. And that's all been validated through collaborations at NIH using laser Doppler techniques. I won't go through the details of that. That's been quantitatively validated. And so now you can take these devices and you can use them on hydrocephalus patients, and we have deep collaborations with the neurosurgery department where I actually have a joint appointment. And we're doing measurements in this particular case on patients who are having their shunts installed or surgically fixed. And so we know exactly what the flow rate is. We put our device there and you can measure whether there's flow or no flow with very high signal to noise. And so to first order, that's good enough. Just yes, no, just a binary. Is there flow through the shunt or not? It'll tell you if your shunt's clogged or not. But you can actually sort of quantitate that flow rate as well using the computational approaches that I mentioned before. So that's a de novo capability. And so because of the tremendous amount of interest that we were getting from patients following the publication of our paper in the popular press that was associated with that, we decided to start a company to get it out of the lab and make it available to patients in need. And so FDA gave us breakthrough designations, de novo. We're in the middle of a pivotal study. It's a small engineering-oriented company just focused on getting the technology out into the hands of patients and physicians. And so we're in the middle of our pivotal trial. I think everything looks good. And this is just a picture of the various pediatric patients that have been part of that trial. And these are the hospitals that we're partnering with for those trials. It includes Stanford as well, and that's a picture of a patient from Stanford's Children's Hospital participating in this study. There's our devices right there, and the shunt's coming down right in that direction. So it's another example. Let me give you one more, and this will allow me to pivot to this second platform that I want to share with you that goes beyond just biophysical measurements, thermal, electrical, optical, measuring motion, sounds, that kind of thing, into a realm of biochemical. And we sort of backed into this. It wasn't an effort to develop new technology for screening for cystic fibrosis, but it turned out subsequently to be very useful for that particular application. But let me just describe what the technology is, how we got started, and what we're doing with this. So if you think about soft electronics, that's kind of a reformulation of a conventional technology, electronics, that in its native form is rigid and planar and incompatible with the soft textures of the body. Same thing could be said with lab-on-a-chip technologies as well, typically made out of glass rather than silicon, but still adopting that simple multi-layer 2D rigid platform. And so we begin to think about microfluidics in the context of skin-compatible embodiments. And we're able to put together a technology that has those characteristics, so soft and skin-like, but now not with a focus on electronic functionality, but instead microfluidics. And why would you want microfluidics on the surface of your skin? Maybe you couple that with penetrating pins, microneedles. We heard a lot about that. Joe's very active in that space as well. But we try to go down that path. There are a lot of challenges in getting IRB approval because of pain risks and infection risks and so on. And so then we thought about like biofluids you could just capture from the skin that don't require any penetration. So unlike interstitial fluid or blood. And we decided to focus on sweat, so eccrine sweat. And so in this case, we have a microfluidic device fully enclosed, but with inlet ports on the backside that couple to the surface of the skin with a dual-sided biomedical adhesive that has openings cut in it, aligned with those inlet ports on the backside. And so in this particular case, then, the natural pumping action of the eccrine glands pushes sweat up through the surface of the skin, through these openings into the inlet ports, and then into a network of microchannels and valves and reservoirs. And you can couple electrochemical sensors using concepts in soft electronics to do biochemical assays directly in that microfluidic platform. But then we got a little bit more attracted to the idea of using colorimetric chemical reagents to do the same thing, thereby eliminating the need for radios and batteries and electronics and reducing the cost structure of the devices. So this was the first example of a device of that type. It has 4 circular reservoirs, each coupled to the skin through a separate inlet port, and then this serpentine structure that runs around the perimeter of the device connected to yet another inlet port. So this is what it looks like, partially filled with artificial sweat. So that inlet port is connecting to this serpentine channel. It's filled with cobalt chloride doped hydrogel, which undergoes a reaction with water that causes a color change from purple to red. So if you just determine the position of the color change front, you can determine the extent of filling of that serpentine channel. And from the cross-sectional dimensions and the contour length, then you can determine the amount of sweat that's been lost from that particular location on the skin. You do that twice, you get sweat rate. So sweat rate, sweat loss coming from this part of the device, and then these 4 circular reservoirs have colorimetric chemistries that respond through a color change to concentration of lactate, chloride, glucose, and sweat and pH. So a simple non-electronic colorimetric device gives you 4 biomarkers and also gives you sweat and sweat rate out of the system. So very, very inexpensive, just a molded piece of plastic. But as with electronics, the mechanical properties become super important, not just from a comfort standpoint and sort of a non-irritating interface to the skin, but in this case, it needs to be watertight as well. You don't want leakage. You lose accuracy in the measurement of sweat rate and sweat loss. So actually, it turns out to be a little bit more challenging, partly for that reason, partly because some of these applications involve vigorous sweat rates that can make adhesion and coupling to the skin even more challenging. So again, it's a problem in combined materials and mechanics. How are you going to combine materials with device layouts that yield mechanical properties that allow skin compatibility and also reduce interface stresses to levels that allow efficient adhesive coupling without extremely aggressive adhesive chemistries. So anyway, I won't go through the details. You can do all of that. This is an example of a device mounted on the outer forearm of one of the grad students who's working on the project. You can see that central inlet port has filled this serpentine channel up to about the 7 o'clock position. And so from that 2 time points, you get sweat rate, you get sweat loss, and it turns out that that local sweat loss measurement can be calibrated fairly well to full body sweat loss. And I'll get back to that in a second because it turns out to be important for managing hydration, due to sweat loss. And then you just sort of take a picture using digital camera. You do color extraction, image recognition. It gives you that color change front, and it gives you quantitation around the color of the 4 reservoirs for measurements of lactate, chloride, glucose, and pH. So that's what it looks like. So you can think about this almost like time-dependent tattoos. To give you an example of how this looks. So this is on my inner forearm while I'm working out in my basement. So I have a device, in this case, just have a color food dye near the inlet port. And so I begin to sweat. It fills up this serpentine channel by an amount determined by how much sweat has been lost from that point. So it gives you a very visual, immediate feedback of how much sweat you've lost at any point during a competition or exercise. So it also turns out to be very motivating. I got up to this point and decided I'm going to try to fill the damn thing, and nearly killed myself doing that up to 6:35. But I was able to fill it all the way. But it's all quantitative. So you actually take a series of pictures like this. You determine the color change front, the fill front at any given time, and then you can generate plots like this. As a cumulative sweat loss as a function of exercise duration. So I'm actually on an elliptical, about constant intensity workout here. But what's amazing is the sweat rate is almost invariant to within measurement precision. It's just running like clockwork, which is pretty cool. And I don't think that's ever been measured. Like very simple measurement, but It gives you some insight into human physiology and the nature of sweat production and sweat release. The other thing that's worth pointing out here is there's really no buildup of sweat in adjacent regions of my skin. And that's because the sweat's constantly evaporating, right? So you kind of lose track just by looking at how much sweat is on your skin of how much sweat you've actually lost. So that turns out to be important. Here's an example of a chemistry for colorimetric measurement of chloride, because this is pretty easy, and obviously it's important to understand the electrolyte level of sweat. Come back to that and its relevance to cystic fibrosis screening. But in this particular case, we're using silver chloranoate, which reacts with free chloride in sweat, more or less electrostatically balanced with sodium. There's a little bit of potassium in sweat as well, but this is giving you a good estimate of sodium chloride concentration. But anyway, it produces this purple anion acidic group with vibrant color change across a range of concentrations of chloride that are physiologically relevant for humans. And so everybody has a different electrolyte level in their sweat that can change with diet, season, health status. But you can determine that concentration to within 2 or 3 millimolars just using colorimetrics. And you can compare that to ICP-AS and as a validation, and that works pretty well. So that's kind of what we were doing. We published a paper, a lot of popular press, and then I get a call from the president of Gatorade, who lives right around the corner from me, actually. I had no idea Gatorade was based in Chicago. But anyway, he's a Kellogg alum, Northwestern alum, very interested in the technology, wanted to know if we wanted to work with them to make a product to provide to their customer base. And the idea is with this type of patch, so determining sweat loss, sweat rate, here's the chloride channel. It will provide information to their customers as to how much Gatorade they need to drink. It's basically the business model. And what type of Gatorade formulation in terms of electrolyte concentration, that's where the X comes in, that matches their particular electrolyte level in their sweat to replenish lost water, but also lost electrolyte. So again, it's sort of forcing function. We can't do that kind of product development through by academic groups, so we set up this company to do that. And then that's been successful. So we produced, 3.5 million of these things. You can pick them up at Dick's Sporting Goods. I have 3 of them. So the first 3 people who approach me after this talk, you get a free sweat patch. But anyway, it's been cool to work with Gatorade. It's just a great company, sort of elite brand. They own 75-80% of the market. But then they also have this stable of celebrity athletes that you get to work with. And so there's Serena Williams. There's a commercial spot involving her with our patch. There's Jason Tatum that was there during the All-Star Game 2020. There's Paul George, like basketball. You can order this off the Gatorade website. But the ultimate was this. So this is Lionel Messi. So I am told he has more followers on social media than any other individual on the planet. So you want to be cool like Lionel Messi, you got to get a sweat patch. Is the way it works. But it's all scientifically validated. So we work with Gatorade Sports Science Institute in Barrington, Illinois. They probably studied sweat more than any other organization in the world, I would say. They've been looking at it for 25, 30 years. Anyway, you can measure the sweat rate, the sweat loss, the microfluidics. There's their standards, just an absorbent pad. Colorimetric chloride, you can compare to HPLC. There's a little bit of scatter here, but there's a point-to-point, spot-to-spot variations, human biology variation, but pretty good there. The interesting thing here is you can also purchase this device in virtual form. If you're interested, if you're a gamer, you've ever played NBA 2K, starting in 21, 22, and 23, you can go in a game and you can buy patches for your avatar. Now, unfortunately, we didn't realize this was a possibility, so this lies outside of our licensing agreement with Gatorade. So we don't get any money. That's actually a commercial transaction that happens within the game, which is pretty cool. But I think the more compelling applications from a commercial standpoint is in worker safety, especially oil and gas. And so we have partnerships with Chevron and BP, and these are sort of industrial athletes. In many ways, more impressive than those regular athletes in the sense that they're adding real value. And we just put a Bluetooth module on top, and so they can get readouts without needing to gain visual access. It's usually under PPE. So why is all of this relevant? It's not health in the traditional sense, but it drives manufacturing and scale and cost, as Joe was referring to previously. So to make this work, make this patch work, we have a continuous reel-to-reel process. It's a 9-layer system, and it's produced continuously at about $1 per patch manufacturing cost. And that has spillover effects on medical applications. So that's where we were. We got a call from pediatricians at Lurie Children's Hospital, and they asked us if they could get access to some of these Gatorade patches for cystic fibrosis screening. I said, what are you talking about? And I had no idea that this is the gold standard for cystic fibrosis, to look at sweat chloride. If it's higher than a certain level, then that's a good indication the patient suffers from this disease. And the way that that's done currently is you have a coiled up tube in a hockey puck type enclosure. You strap it to the surface of the skin, sweat fills the tube, you pull the tube out, you suck the sweat out, and then you put it into a chlorimeter. It's a benchtop analysis machine. And that has problems. It's expensive, it requires a technician, you can only do this at a hospital, and you end up with a 25% failure rate due to insufficient sample collected because of leakage. And so the idea was just take that patch and just modify it. It was a little bit softer, a little bit thinner, a little bit smaller in terms of the reservoirs, but now we just put the sticker on, you collect a tiny amount of sweat in these reservoirs. It's good enough to do the colorimetrics, you get the chloride, and you do the screening. So it turns out to work really well. We've done a lot of studies on pediatric patients. We're very excited about it. This guy doesn't look like he's fully convinced on the technology, but it's really fun to work with these kinds of patients. Really amazing, and they're really responsive to the appearance of the devices. You give them a device that's just a white thing; they're not interested in it. I don't know if you can see this. You put a sticker of Lightning McQueen on top; they can't get enough of them. And so maybe some insights into design for use with adults as well. The kids will give you uninhibited feedback immediately, so you can really understand what's going on. Anyway, it's fully validated, and that works. Published in Science Translational Medicine about a year ago. So let me just conclude with this kind of notion. So why are you interested in sweat for cystic fibrosis screening is pretty clear. Sweat's a relatively underexplored biofluid, though, and so it really requires a lot of research to understand species concentration in sweat and how that relates to blood. But there might be some interesting opportunities of pediatric nutritional health in measuring nutritional biomarkers in sweat. And so we've developed colorimetrics for doing vitamin C. It's present in sweat. You can measure that over a physiologically relevant range for kids. Calcium, we could do that as well. Zinc, iron. So a number of different nutritional species. You can detect them in sweat, and you can use that information to determine nutritional balance. So this is the basis of a new program that we're discussing with the Gates Foundation. But in order to compare to blood, you can do these kinds of studies. So you can measure those species in sweat as a function of time after you take a supplement, either a vitamin tablet or a vitamin patch. And you can look at the kinetics of how long it takes for the concentrations of those species to peak up in sweat and how long it takes to decay back down. And you compare that to those two time constants associated with previous studies of these species in blood. And it looks like they correlate pretty closely, but this is very much ongoing work just to give you a sense of where things might go into the future. And so you could develop a patch that has multiple analysis chemistries. You do nutrition, you do chloride, you can do other things, and really scale this in an important way to do a broader, more comprehensive biochemical assessment of health status. So I think I'm out of time, been out of time maybe 2 or 3 minutes. I apologize for that. But anyway, I described some work in this kind of skin-like electronics, skin-like microfluidics with a focus on translation, try to give you a sense of what we're trying to do with these technologies to address patient needs. But all the work is very collaborative, so we work with a number of folks in engineering science. It's really that interface to the clinical medical community that's so important for us. I want to acknowledge those folks, but the most important people are the students and postdocs who do the work. This is a picture of the group from holiday party here in December. Give you a sense of what the team looks like. So thank you very much for your attention. Be happy to answer questions.
Jack Mormon. So this is terrific, of course, and very impressive. And I'm interested more in the other extreme. You showed neonates and, of course, the other healthy arms that you were using. The more difficult might be seniors with dry skin, with fragile skin that tears, all these other. Are you working on that? And if so, where are you on that?
Yeah, we are. I didn't embed that in the talk. We work with Parkinson's patients, Alzheimer's, dementia patients, stroke survivors. Aphasia and dysphagia are two conditions that we're looking at in those patient populations. So, I mean, all this is kind of new for me, but swallow is a big deal, and being able to measure swallows when they happen relative to the respiratory cycle. If it doesn't happen in the appropriate place, you can choke yourself, and this becomes an increasing problem. People with dementia and Parkinson's, so we work with speech pathologists. We also work with a rehabilitation specialist at the Shirley Ryan Ability Lab in that context. Aphasia as well, stroke survivors relearning how to speak and using these kinds of devices to measure vocal activity and vocal cadence and frequency and that kind of thing. So these are all devices that mount on the suprasternal notch, so we can track when swallows happen relative to the respiratory cycle. We get in chest wall movements. So we have deep interactions and large funded NIH programs in those areas. I think the skin interface challenges are more significant with premature babies, however, because there the skin is highly underdeveloped. It's not really even like skin. It's almost like a paste. It's almost like an open wound. The epidermis and the stratum corneum is not even fully formed. So I agree there are challenges in both classes of patients. I would maintain it's most challenging with the premature babies. But I think a lot of the loose skin causes problems, not so much in damage to the skin during application and removal, but just in sort of mechanical isolation of the devices from the body. You're sitting on a loose, decoupled surface, and so the signal quality can be impacted by that looseness. That's more of a challenge with the elderly patients. Yeah.
Thank you for the great talk. I have a question about the power management. What is the source of the power and how you manage the power over time for continuous management?
Yeah, so great question. So the microfluidic devices no power, so they're self-powered by the eccrine sweat glands, I guess is maybe a way you could think about it. So there's sort of two of these skin interface electronic platforms that I kind of skipped through pretty quickly. The first and the most aggressive is this full epidermal type device where skin-like properties. That was the vision. We were able to do that. That's these devices. So there's no battery. So they're being powered by magnetic inductive coupling between, in this case, it's a transmission antenna that's sitting in the base of the chair, but also would be placed underneath a mattress in the isolette. And so it's operating at RFID tag standard frequencies, HF 13.56 megahertz. And so it's an oscillating magnetic field that couples to this coil here through inductive coupling to produce power locally on the device. And so we can do maybe 5 milliwatts that way. So it's a very low power radio that's using the same magnetic inductive link for data transmission. And that works really well, provided that you're within about a half a meter of the transmission antenna. It's like the antennas that sometimes libraries you go out the door. It's the same technology basically. So we have those coils, the transmission coils running about 8 watts, and we're harvesting about 1/1000th of that power, 8 to 10 milliwatts, and that works pretty well. That is not applicable to health clinics in Kenya, Zambia, Ghana, some of these places, because there's no reliable source of wall plug power for the transmission antenna. So for those applications, I mentioned that we had to make some engineering modifications to address the interests of Gates Save the Children, and we're interested in this too. So you have to have onboard power. It's the only way to do it. And so here we have very small lithium polymer batteries that are wirelessly rechargeable. It's about a 30 milliamp hour battery, roughly. And the other thing is that we can't use that inductive link for data transmission because the reader's too expensive, and you can't afford to have a separate computer associated with each of your monitoring devices. It has to communicate with the phone, because phones are ubiquitous, smartphones, even in Zambia. So it needs to be a wireless link that's compatible with standard cell phone technology. So we had to go to Bluetooth. It's more power hungry than that NFC approach, but that was the requirement. Because the cost structure here, just to sort of orient you, is measured in the cost per patient monitoring day. That's the metric. And so for use in these particular parts of the globe, it's a few cents per patient monitoring day. If it's much more expensive than that, it's just not affordable, and nobody's interested in it. So we had to go through a detailed costing assessment to sort of justify that this could work. So that was reviewed, as in great detail. But those are features that needed to be present, and the consequence is these are not as skin-like as those other devices, because they have to be reusable. It's the only way that you can get a few cents per patient monitoring day. These devices have to be used 600-700 times. So you can amortize out of the overall cost structure the cost of the device. It basically disappears when you use it 500, 600, 700 times. So anyway, long answer to your question. The thin devices designed initially for Lurie Children's is fine because all of the monitoring equipment used in NICU facilities in the U.S. are single use. They don't try to sterilize them and reuse them. And so the idea of a patch, you put it on, you peel it off and throw it away, that's very much compatible with the way patients are cared for here, but not in those parts of the globe. So there's two separate platforms.
John, thank you, John.